Hi, my name is Eric Patton, and today I will be presenting the endosymbiosis theory and the endogenous theory and the origins of mitochondria and chloroplasts in eukaryotic organisms. I drew kind of a diagram here on the stages of the cells throughout basically billion years of evolution, and here we start with ancestral eukaryotes that depend mostly on their energy in the plasma membrane of the entire cell. And the DNA just kind of hangs out here on the plasma membrane. The formation of the organelles in the cell start by enfolding with the plasma membrane within itself into the cell, forming these small pieces of phospholipid layers that begin to form structures inside the cell, coating this DNA eventually, and compartmentalization formation starts and begins to form ER, which is your endoplasmic reticulum, the nucleus, and contains the DNA. And that just covers the endogenous part, everything starting in an internal cell and then forming those organelles just with the cell membrane alone. And if you go further to go into mitochondria, those are eventually acquired by endocytosis of non-sulfur purple bacteria. And we'll go over that a little bit later on how we know this. And the endocytosis of the purple bacteria results in the double membraned structure of the eukaryotes plasma membrane and the purple sulfur bacteria's membrane. And that's where you get two membranes from two completely different organisms, and eventually the formation of mitochondria. And eventually, because ATP is no longer formed on the outside of this cell membrane, the ATP that's used from the bacteria's cellular membrane is used to produce ATP in what we know as modern eukaryotes today. And here's a little bit of an explanation between the endogenous theories. The stages of the membrane are enfolding within each other to start compartmentalizing the organelles. Continuous folding forms the nucleus, ER, Golgi apparatus, etc. ATP and lipid synthesis role went to mitochondria and chloroplasts, so the plasma membrane has lost its function as a producer of ATP and now is dependent on the mitochondria and chloroplasts of modern eukaryotes. And then genes from the host nucleus are transferred to the mitochondria and chloroplasts, and this is just the endogenous theory. So there's more proof in the endosymbiosis theory that mitochondria came from bacterial organisms. Instead of having the nucleus transfer DNA to an organelle, it actually used the genome inside of the bacteria to produce ribosomes, proteins, and ATP, etc. And then mitochondria and chloroplasts are acquired by endocytosis in the endosymbiosis theory of prokaryotes, and specifically aerobic bacteria, oxygen-dependent prokaryotes. The double membrane consists of both the host membrane, which is highlighted here in black, and the endosymbiont membrane, which is highlighted in purple, the purple bacteria. And we also know that this is possibly a legitimate theory because the mitochondria, as well as the host cell, undergo binary fission by reproducing themselves. So it's almost like there's another organism inside another organism. And so they communicate with each other to know when to replicate through proteins and catalytic processes. And then the ultimate fact we can say is genetic testing proves that mitochondria and chloroplasts share DNA with non-sulfur bacteria and cyanobacteria, respectively. So mitochondria tested for very similar genomes with non-sulfur bacteria, and chloroplasts test with cyanobacteria in their genes with chloroplasts. And that concludes my explanation on the differences between the endogenous and the endosymbiote theory and their origins of mitochondria and chloroplasts. Have a good day.